Okay, so this week we have to study about dosage compensation, GIST, and non-coding RNA in imprinting. So this week we have a lot to cover because we had 7 lectures. So we'll try to quickly go through the theory and then we'll try to solve the questions. So there are many examples, especially in the last 2 lectures, in lecture 6 and 7. So we'll not be discussing each and every example because then it would take a lot of time for us. So we'll try to just summarize each and every lecture and have a gist of each and every lecture. So like this, we'll be moving. So our first lecture is the role of YY1 protein. Okay. So basically, YY1 protein is required for gist localization. So yeah. Hmm. So this is YY1 is a protein which is also known as yin yang 1, and this protein is required for the localization of the cyst. So this protein helps in tethering the cyst onto the chromosome, the place where it is produced. Okay, so it is required for localization of the cyst. So if you use YY1 siRNA to block its translation, then you will not have the ability of cyst localized on the corresponding chromosome. So you can see over here in this figure that this is the DNA, cyst DNA, and this is the YY1 binding. This is the YY1 binding domain in the cyst RNA. This is the YY1 protein, and this is here it binds. It acts as a linker also. It acts as an adapter also. So cyst will be able to attach to its target, that is to the X inactivation center, with the help of the protein called YY1. So we have already discussed a little bit about the YY1 in the previous lecture. So I won't be repeating it. Just to recapitulate, this YY1 is basically required because it helps cyst to attach to its targets. Okay, so cyst will be able to attach to its targets, and which is what? Which is the X inactivation center with the help of the protein YY1. So this YY1 helps the cyst protein. Then there is a paper where there is a paper that sir has mentioned, and this paper actually tells us that the mutation, so xi means inactive X. So in the previous class we have studied about dosage compensation. We have studied that in females, one of the X is inactivated so as to equalize the expression of X chromosome. So in the case of humans, in the case of Homo sapiens, if there is XY is male and XX is female, so one of the X is inactivated. Okay, and this inactivated X is known as XI, which is the inactivated X. So what this paper says is that the inactivated X in female accumulates somatic mutations during tumorigenesis. What does it means? That this inactivated X chromosome here, mutation rate is very high. Okay, so the mutation rate in the X chromosome is very high, but these mutations are not repaired during replication because the replication of this inactivated X chromosomes occurs very late. So these X chromosomes have a very late replication. That is why these mutations cannot be repaired. Okay. So what we get to know from here is this is the inactivated X chromosome, and mutation rate is very high. But we know that mutations can be repaired in during replication by the process of DNA repair. Okay, but these mutations are not repaired. Why? Because these inactivated X chromosomes have a very late replication. Okay, so now you can see over here. Just a minute. Some students want to write some messages there. Okay. Hmm. See here, this is a normal somatic cell and this is a cancer cell. So you can see that in a normal somatic cell, there are some mutations in the X chromosome, but in a cancerous cell, the mutations in the X chromosome is very high. That means highest number of mutations in a cancer cell occurs in the X chromosome. And even if we compare the two X chromosome, I mean in the X chromosome there is an inactive X and there is an active X. So you can see that the mutations in the inactive X is the highest. Okay, so this is what occurs. That mutations in the inactive X chromosome in the case of cancer cells is the highest. But these mutations cannot be repaired. Why? Because this is an inactive X chromosome, and because it is an inactive chromosome, its replication occurs very late. That is why these mutations are not very much repaired, but they do not have that much of deleterious effects because this X chromosome is already inactive. Okay, this X chromosome is already, I mean, it is not expressing. So whatever mutations are also there, they will also not expressed. So it is not so much of, it's not such a problematic situation for us, but we need to know that in the tumor cells, most of the mutations occurs in the inactive X chromosome. Then we come to the next point, which is genetic imprinting and the role of shRNAs. So about genetic imprinting, we have studied a little bit in the previous class also. What is genetic imprinting? It is a process which results in expression of only one of the alleles of a gene, while the other allele originating from one of the parent is silenced. So we all know that for every gene, for most of the genes, there are two alleles. Suppose if a gene which corresponds to um, right, so it will have two alleles, one and two. Okay, so this is a normal example that I'm giving you. Suppose if one of the allele is coming from one of the parent, other allele is coming from other parent. Genetic imprinting means that it is already imprinted that for the expression of height gene, the allele from mother will only be expressed. The allele from father would not be expressed. It would be silenced. So it is already imprinted. It is already decided that one of the allele will be expressed and other of the allele will be silenced. This is known as genetic imprinting. Okay. So one of the expression is only from one of the allele while the allele originating from the other parent is silent. Okay. Then the differences of expression from both alleles are due to states of chromatin and also to different methylation status of the DNA. What does it mean? It means that if I say that here this is getting expressed and this is not getting expressed, that means this would be methylated or demethylated. Tell me, anybody, if I say that this small h is not getting expressed, then what should be the methylation status of this HLE? It should be methylated or it should be demethylated?
Methylated.
Methylated. Yes, because methylation, it will repress transcription. Then this is okay. Then whether this area, whether this allele will come in euchromatin region or heterochromatin, what it should be? Euchromatin or heterochromatin? Anybody?
Euchromatin.
Euchromatin. Okay. But okay, so do you know the difference between euchromatin and heterochromatin? So this is the portion which is expressed. Okay. Euchromatin is the region of the DNA which is getting expressed, and heterochromatin, it is the region which is
Express.
not getting expressed. Okay. So if I say that this H is imprinted, I mean it is not getting expressed, then it should belong to euchromatin region or heterochromatin region. Hmm? Anybody?
Heterochromatin.
Heterochromatin region, yes. So this is what they are saying, that the difference of expression from both alleles are due to different states of chromatin. That means this is because it is already decided that there is genetic imprinting. Only one of the allele will be expressed, other of the allele would be silenced. So the allele which is silenced, it will have a greater methylation and it will belong to heterochromatin region also. This is what genetic imprinting is. Now, genetic imprinting is helped or it is undertaken by a class of RNAs which are known as small heterochromatic RNAs or shRNAs. Okay, so these small heterochromatin RNAs or shRNAs, what are they? They appear to be essential for establishing and maintaining the imprinted status of gene. So basically, this shRNA, it helps in maintaining the genetic imprinting. It helps in maintaining the imprinted status of the gene. But this is not the only function of these shRNAs. These shRNAs have different roles as well. But one of the roles in context of what we are studying genetic imprinting is that this genetic imprinting is facilitated with the help of small heterochromatic RNAs, that is shRNAs. Okay. So up till now, we have studied about the role of YY1, and then we have studied that this inactivated X, which is present in females, it harbors most of the somatic mutations, and these mutations cannot be repaired because of its late replication. Then we studied about the genetic imprinting. What is genetic imprinting? What are the characteristics of the silenced genes? They are highly methylated and they belong to the heterochromatin region. And lastly, we studied that shRNAs are essential for the genetic imprinting to be maintained. Then we come to next thing. So we are studying about shRNA only. So basically, there are two kinds of models for for the DNA models for the recognition with the help of shRNA. One model is known as a DNA recognition model, and the other model is known as an RNA recognition model. Now, what does it mean? If it's a DNA recognition model and if it is an RNA recognition model, with the help of this figure, what can you tell? What would be the difference between the two models? Anything that you can um, assume by seeing just the figure and the name? It's a DNA, it's an RNA. What should be the difference? Anybody who wants to try? What would be the difference between a DNA recognition model and an RNA recognition model? Hi, Supreja. Do you want to attempt? Kanchana, Tarkeshwari, Yuvranjani. Anybody who can think of any answer? So, what is the difference between DNA and RNA? Anybody?
It's double helix DNA.
Okay, in the term of expressivity, which portion is expressed? No? Okay, fine. So, if this is the whole genomic region, okay, which has introns, exons, introns, exons, then when it is transcribed and when RNA is formed, we only have the exonic regions, E1, E2, E3. We only have the exonic regions. That means we only have those regions which are getting expressed in mRNA. So we have the expressed regions. That means RNA contains the regions which are going to be expressed, whereas DNA has both of the regions which are going to be expressed also, which are not going to be expressed also. So this is what is the difference between the two models also, DNA recognition model and RNA recognition model. So what is there in both the models, similar pattern is observed, except in DNA recognition model, there is no need of gene expression. See, in DNA recognition model, there is no need of gene expression. It can be imprinted because of shRNA coming from elsewhere, binding on the DNA strand and triggering the silencing event. That means, see, genomic imprinting or genetic imprinting is facilitated by shRNA. This is what we have just now learned. Now, this shRNA has to come and it has to bind to the genomic region or it has to bind somewhere so that it can start the genetic imprinting process, correct or not? So, it can bind in two ways. And these two ways are the two models. First is the DNA recognition model, and the other one is the RNA recognition model. That means this shRNA which is coming, which is now it is going to do the genetic imprinting process. When it binds via DNA recognition model, it just comes, sit, and do the silencing event. There is no need of the expression of the DNA. But if it follows the RNA recognition model, then what has to be done? The gene which has to be silenced, it has to be expressed. Here, there is no need of expression. Here, it has to be expressed. So the gene has to be expressed for a while, and then this will be triggered by shRNA and it will lead to silencing through the silencing complex. That means whatever gene which is going to be silenced to maintain the genetic imprinting, it first needs to expressed for a little while and then only it can be silenced. Whereas in this model, the gene which is going to be silenced, which is going to be imprinted, it need not to express. shRNA will directly come, bind to that gene and silence it. Is it clear to everybody? Are is the difference between the two models clear to everyone? What I'm trying to say here? So basically, shRNA has to silence the gene in order to maintain the genomic imprinting. Because what is genomic imprinting? Genomic imprinting means only one of the allele is getting expressed. Okay. So when we talk about genetic or genomic imprinting, we are saying that if there are two alleles for a gene, one of the allele is silenced. That means the expression is coming from only one of the gene. Now, as one of the gene is silenced, it has to be silenced through some mechanism, and that silencing is mediated or that silencing is helped with the help of shRNA. Okay. Now, how does it do that? This shRNA has to come and recognize the gene which it has to silence. Okay, it has to recognize the gene which has to be silenced. Now, this recognition can be done either with a DNA recognition model or with a RNA recognition model. If it follows a DNA recognition model, there is no need of the gene to be expressed. So, if it is a DNA recognition model, no need of gene expression. If it is a RNA recognition model, the gene has to be first expressed for a while and then it will be silenced with the help of shRNA. So here, gene is expressed for a while. Okay. So is this clear now? Now is it clear to everybody?
Yes, ma'am.
Okay, I'm unable to hear the voice, but I am assuming that it is clear to you. Okay. So this we have studied about regulation, transcriptional regulation we have studied. Now we are going to study about translational regulation. So translational regulation with the help of non-coding RNAs. So what we are studying, our subject is right. See, always try to connect the dots. What we are studying from the first class itself. So we are studying about RNA biology. Okay, in RNA biology, we have studied about different types of RNAs, what are their functions. Then we have studied how this RNA is uh, processed or modified after the transcription. So we have studied about post-transcriptional modifications. There are some basic general post-transcription modifications, and there are some unique post-transcription modifications then that we have studied. Then we studied that apart from the RNA, such as mRNA, which is mostly studied, which is responsible for the formation of proteins. So mRNA will only form proteins. So mRNA codes for proteins. There are some other RNAs that does not code for proteins. Okay, but they have some other functions. And what are those RNAs? Those RNAs are like tRNA. Okay, then you have rRNA and other non-coding RNA. So this tRNA and RNA also it is very well studied. But other non-coding RNAs means you have miRNA, then siRNA, lncRNA, and many more. So these are the different non-coding RNAs that does not code for a protein, but it does not mean that they are junk or that they do not have any function. They do are very important for the system, but it's just that they're not making proteins, they're doing some other work. And what are those other works? One of the other work that is achieved by this non-coding RNA is genomic imprinting that we are studying today. Okay, genomic imprinting. And in the last class, we studied that one of the work which is done by these non-coding RNA is X chromosome inactivation, or in a broader sense, dosage compensation. So that is also one of the work that is done by non-coding RNA. And today, what we are studying, genomic imprinting, this is another kind of work that is done by non-coding RNA. So not only that they help in genomic imprinting, they are also involved in targeting certain mRNA molecules and silencing it, which is known as RNA interference, which is RNAi. So, does anybody knows anything about RNA interference? Any idea what RNA interference is? Any basic idea? Think nobody has any idea about RNA interference? So, from the name only, you can guess that the name is RNA interference. That is, it is something it is interfering with the RNA. There is some interference in the action of RNA. I mean, there is some interference in the expression of RNA or the work that is done by RNA, maybe. So, this is achieved by non-coding RNA. So, we'll see what is RNA interference. I think we have already studied a little bit about RNA interference in our previous classes, but we'll try to study it in more detail. So, mostly RNA interference, what does it do? That RNA interference, it will silence the expression. And there is one example which I have not mentioned here. I think that example was just before this slide, where they have shown that how RNA interference can silence the expression of genes. But this is interesting and it is new that RNA interference not only silence the expression, but sometimes it can also activate the expression. And that is why this example becomes important and different. Because we all know that RNA interference is required or is the process which achieves the silencing of the expression of a gene, which means the translation is blocked, the protein is not formed. But here in this example, we'll see that RNA interference is helping the expression of the protein or helping the expression of the mRNA. How it does? This is the this is one mRNA which is RPOS mRNA. Okay, this RPOS mRNA has a ribosome binding site. So here the translation will take place from this ribosome binding site. But what is the problem here? That this RPOS mRNA has this stem loop structure. This is the stem loop structure. Okay, it looks like this is a loop and this is the see. This portion is the loop and it looks like a stem. So it is known as a stem loop structure. And why this stem loop structure is formed? Because there is what is the how stem loop structure is formed? Tell me, anybody who can tell me how a stem loop structure can form within an RNA. Anybody knows how a stem loop structure is formed within an RNA? Nobody knows, or nobody is even listening. Okay, fine. See, okay. So this is RNA. It has A, T, G, C, T, T, A, whatever it is. This is another RNA. This
Hairpin
RNA
loop,
can pair with this, sorry,
hairpin loop pipe.
hairpin loop. Yes, hairpin loop. That is also stem loop structure only, correct? So you see, this is your RNA, and this RNA can pair with this RNA when the sequences are complementary. That means uh, it should be U because it is an RNA. So if it is U, A, C, G, A, A, U, U, then this RNA can pair with this and it will form a double-stranded RNA. Now, suppose if in this RNA, this region is complementary to this region, then it will bind, and when this will bind, it will eventually loop out like this because this region will bind to this region, so it will loop out this. So you, it is like a hairpin. So we use U pins. These kinds of U pins we use. This is like a hairpin structure or a stem loop structure because it's like a stem and it is looping. So that is what they are saying, that in this RPOS mRNA, this is the ribosome binding site of this RPOS mRNA, which helps in the translation. But this RPOS mRNA, because of its base sequence, forms a stem loop structure. And because this stem loop structure is formed here, this ribosome, it cannot move this side. See, if there is, this is the stem loop structure and this is your ribosome, it will move like this and then it cannot go like this. It has to move straight only. So ribosome cannot move or cannot pass this stem loop structure. So if somehow this stem loop structure is released, then ribosome can move and then translation can happen and then expression would be there. So what happens that this is your non-coding RNA, which is dsRA RNA. So what it does is that it comes, see, the ribosome binding site is masked because of this stem loop secondary structure. Okay. So RPOS RNA, which is a sigma factor RNA, if it is present does not mean it will be translated. It is by default. Okay, just leave it. See, this RPOS, this portion, it will come and it will bind with this structure, this RNA. This RNA, this whole region, this will bind with this. So it is getting straightened up, and this is also straightened up. It is will bind. So there will be no secondary structure formation. So this double-stranded dsRA RNA, it will come. It will bind with the RPOS mRNA, and hence it will prevent the secondary structure formation. And then the ribosome binding site is accessible, and the translation can happen. Okay. So what you have to understand is that this RPOS mRNA has a tendency to form this secondary structure. But Before this RPOS mRNA can form this secondary structure, this DSRA RNA will come and it will bind with this. So before it can form secondary structure, it will form like this, a double-stranded structure. So there is no blockage of the ribosome binding site, and your translation can happen. So this is how a so normally we see that a non-coding RNA, it comes and it silences the expression, but here it is binding with this RPOS mRNA and it is helping the preventing the RPOS mRNA to form this secondary structure, and in a way it is helping in the expression. It is helping in the translation. Okay. So is this clear to everybody what I'm trying to tell? This mechanism, this method is clear? Okay, I assume that it is clear because I cannot see much of the response. That's fine. Now, so we have studied that how RNA interference can regulate the translation process. So it can regulate the translation process positively also. It can regulate the translation process negatively also. Negatively means it is suppressing the expression. Positively means it is helping in the expression. And this is the example of positive regulation. Okay. Now we come to next thing, that is protein function modulation. What is protein function modulation? So some non-coding RNAs, it can bind directly to proteins and alter their structure, enzymatic activities or ligand binding. Okay. And the targets of such non-coding RNAs are proteins which are involved in transcription. So basically, we know that non-coding RNA, it comes and it binds to RNA and then it silences the expression. Mostly, we have also seen one example of uh, mm, where the translation is getting positively regulated. It is getting upregulated. But we don't, what protein function modulation says, that non-coding RNAs, they not only bind to RNA, they can also bind to proteins. Again, this is new. We know that non-coding RNA can bind to RNAs and then modulate or regulate its expression. But here it says that non-coding RNAs can also bind to proteins. And when they bind to proteins, they alter their structure, they alter their enzymatic activities, and they alter their ligand binding. Okay. Now question comes that what kind of proteins will these non-coding RNAs bind? So these non-coding RNAs will bind proteins which are involved in transcription mostly. So basically these non-coding RNAs, it will bind to proteins that are involved in transcription. Fine. Now, so this was lecture number 2. So I hope that lecture number 1 and lecture number 2 are clear to you. It is simple only. Just you have to, you should know the concept. And then in the last two lectures, you have a lot of examples and number of examples. If you know the concept, then you can go through hundreds of examples also, no problem. But you should know the logic and the concept. That is the most important thing. Okay. So as we were studying about RNA interference, we'll continue to study about RNA interference. This we have studied previously also. In last week also we studied about, we dealt about RNA interference and how this non-coding RNA is processed and it is made, and then how it actually does its function of silencing the expression of genes. So here you can see that how does silencing work. This is your double-stranded RNA. So double-stranded RNA, dicer acts on this double-stranded RNA. So first thing is digestion. So what happens? Digestion of this double-stranded RNA, and then it forms siRNA. Okay. Once this siRNA is formed, it goes and it unwinds and it forms the RNA-induced silencing complex. So this is your RNA-induced silencing complex in which siRNA comes and bind. So siRNA unwinds, it comes and it binds to the RNA-induced silencing complex. So first step is digestion. Second step is unwinding. Then what happens? Then this siRNA will bind to the mRNA, which has to be silenced. So third is binding. And afterwards, it degrades that mRNA. So fourth is degradation. So this is one, two, three, four. Digestion. Digestion of the RNA to form the non siRNA. Unwinding so that it can bind to the risk complex. What is risk complex? RNA induced silencing complex. That means the complex which is required for the silencing of the mRNA. This is important. Remember the name. Then binding, binding with the mRNA, and then there is degradation of the mRNA. So what is this clear? This RNA interference process? Is it clear to everybody?
Yes, ma'am.
Okay. Um, so Jahanara, I think I'm unable to hear your voice. It's fine. So if it's clear to you, you can just type Y, or if it is not clear, then you can type N. Because I'm unable to hear the voices actually. I hope my voice is coming. Okay, fine. It's clear. So this RNA interference, this is the process by which RNA interference is achieved. Fine. Now comes what are the functions of this RNA interference? Why mRNA is silenced? We all know that from DNA replication, when transcription happens, it is an energetically expensive process. You know, when you have to do transcription, when a cell has to do transcription, it has to give its time and it has to give its energy. So why to waste such so much of energy and time when it has to be degraded? So why RNA interference is there at the first place? Why it will degrade RNA which is made by spending of some amount of ATP. Why? Because this RNA interference acts as a defense against double-stranded RNA containing viruses. That means suppose if a cell is infected with a virus, some outside virus has infected cell, because our system has this RNA interference technique or this mechanism. So what will happen that our system will degrade the viral RNA. So the viral RNA is degraded in this way. So it will provide a defense mechanism against the double-stranded containing viruses. Okay, this is the first thing, that is defense against double-stranded RNA containing viruses. What is the second thing? It stabilizes the genome by sequestering repetitive sequences such as mobile genetic elements. That means if there are a lot of repetitive sequences present in the genome, then that is also a problem. If there are a lot of mobile genetic elements, then that is also a problem. So can you tell me what are the mobile genetic elements which are present in our body? What is the name of that? There's a term for mobile genetic elements. Can you tell me what is that term? Mobile elements. What are they called as? Think nobody know? Yes, Yeshwari, you wanted to say? Tell me.
Transposable elements.
Yes, transposon. Very good. So Transposomes are the mobile genetic elements, and we have already studied that these transposomes are these transposomes can cause mutations also. So, activity of these transposome needs to be controlled. And for controlling the activity of the transposome, this RNA interference comes into picture. So this RNA, because of this RNA interference mechanism, so highly repetitive sequences and transposomes, they are degraded. So they maintain the stability of the genome. Then control cellular development. Dicer knockout mice do not survive past gastrulation. So that means, so when in cellular development, we all know that there has to be a limit up till which a gene has to be expressed and an organism has to grow. So you cannot have fingers which are growing for an indefinite period of time. It has to stop. The growth of fingers has to stop at a particular point. Similarly, the growth of each and every cell has to stop at a particular point in order to make a perfect, stabilized, and a proportionate body. Okay, so that means the expression has to stop at some point. So one of the ways of stopping the expression is RNA interference. So these are the three main important roles of RNA interference. So we can say that RNA interference has a lot of applications, and it indeed is important for the proper cellular metabolism and cellular development of our body. Now, there are two things. RNA interference is carried out with the help of siRNA. And we have also studied that microRNAs, that is miRNAs, they also silence the gene expression. Okay. So miRNAs, they also silence gene expression. Okay. And siRNAs, they are doing RNA interference, and they are also silencing the gene expression. So now comes that what is the difference between the mechanism by which miRNA does the silencing and siRNA does the silencing with via RNA interference. So major difference is, see, whole of the mechanism is same only. That dicer will act, it will form either miRNA or siRNA. Then either miRNA or siRNA, whatever is formed, it will go to the risk complex and then it will do the function. But the major difference or the most important difference between microRNA and siRNA is that microRNA does not have a complete or a perfect base pairing with the RNA, whereas siRNA has a perfect base pairing with the target mRNA. That means whatever mRNA that has to be repressed, siRNA does the perfect base pairing, whereas miRNA, it has an incomplete base pairing. Okay, this is very important. I'll give you 1 minute to read this and then I'll explain. So now I hope you read this. So you see, when siRNA, that is small interfering RNA, they have perfect base pairing. So when they have a perfect base pairing, what happens? The target mRNA is cleaved. But when there is incomplete base pairing, you see, this portion is not paired with the microRNA. So then there is no cleavage, but the translation would be repressed. So these 2 points are important. One, two, one, two. That siRNA, it does a perfect base pairing, which leads to the cleavage of the target, whereas miRNA, it has incomplete base pairing, which leads to translational repression. So miRNA cannot do the cleavage of the target because pairing is not perfect. Okay, so this is the major difference between the mechanisms of microRNA and siRNA. Now, another, it is also very interesting and important. I mean, interesting as in that whatever generally we study about microRNAs or about RNA interference, this is something which is different from it. So, all we know is that RNA interference is a post-transcriptional control method. Okay, that after your transcription happens, when your mRNA is formed, then this mRNA is silenced. Either it is cleaved with the help of siRNA or translation is repressed with the help of miRNA. But now comes the question that is it always a post-transcriptional control which happens in the cytoplasm, or it happens in the nucleus also where it is blocking the transcription itself? So, although this is a very common view, it does not always have to be the case. That means siRNA can be transported to the nucleus and act as shRNA to block the transcription. This is very important. Okay, what does it mean? We know that non-coding RNA has a role in genomic imprinting. That is what we are studying. We studied about shRNA. We studied how shRNA helps in genomic imprinting. So shRNA, it binds to its target gene and it blocks the expression of the transcript, that particular gene. It blocks the expression. So that means these shRNAs are nothing but the siRNAs that has been transported to the nucleus. Interesting. Remember, so we talk about siRNAs, okay? And these siRNAs are the ones which are involved in cleavage of the target mRNA. That means mRNA is already formed. It has come out of the nucleus, it is in the cytoplasm, and now siRNA is coming and acting on this miRNA to cleave it. But this is not always the case. This siRNA can go from the cytoplasm to the nucleus and then it can block the translation or transcription itself. And when it is inside the cytoplasm, it acts like shRNAs. Just like shRNA, it blocks the transcription and silence the gene. So in a similar pattern, in a similar manner, this siRNA also goes inside the nucleus and it blocks the transcription. So we can say from here that RNA interference is not only limited to cytoplasm, it can also happen in some cases inside the nucleus also. So it is not always a post-transcriptional control. It can also block the transcription itself. Okay. I hope this is clear. Is it clear to everybody? Okay, my voice is breaking. So is my voice, I mean, clear now? Can you hear my voice now? Or is it still breaking? It's fine. Okay. So if you weren't able to understand any point because of the some network issue, you can just tell me. I can repeat that. Fine. Otherwise, I'll continue. Okay. So is it clear to everybody what I was trying to tell you about RNA interference, the difference of miRNA and siRNA? Is it clear to everybody? Okay. I assume that it is clear. Ma'am, repeat. Okay. So which portion you want me to repeat? This one more time. Okay. So I'll just quickly repeat it that we are talking about RNA interference. Okay, so RNA interference means that we are interfering the expression of RNA. And this RNA interference is mediated by siRNA. Okay, now we have already studied that silencing of the RNA can also be achieved with the help of miRNA. So miRNA also silence the genes and siRNA also silence the gene. Now comes the question that if both are doing the same function, is there any difference between their activity? So yes, there is a difference. Although they are made in the same manner, that means dicer acts on it and then they go into the risk complex and then they are matured, and then they act on the target RNA, but their action is different. What is the difference? This siRNA, it can completely and perfectly bind with its target mRNA, whereas this miRNA, it cannot perfectly bind with the target mRNA. You can see here, there is a perfect base pairing that is happening, but here the base pairing is not perfect, there is incomplete. So because siRNA can have a perfect base pairing, so they can cleave this miRNA. This miRNA can be cleaved. Okay. See, this miRNA can be cleaved. Sorry, mRNA can be cleaved. Whereas because miRNA, they do not have a perfect base pairing, they cannot cleave the target mRNA, but it will repress the translation process. So this is the difference between miRNA and siRNA. Next, we come to that we say that siRNAs, it cleaves the target mRNA. That means since it is cleaving an RNA molecule, since it is cleaving an mRNA molecule, that means that if this mRNA is already formed, then only it is cleaving it. So there is a question: can RNA interference happens uh, before the mRNA comes out of the nucleus to the cytoplasm also? So the answer is yes. So just like shRNA, we know the function of shRNA. We have studied over here. See, we studied. Just a minute. Yeah, here we have studied that shRNA, it is small heterochromatic shRNA. They are required for the silencing of the gene and they have a role in genomic imprinting. So it's similar to shRNA. What happens? Then these siRNAs, which are normally present in the cytoplasm. Remember, siRNAs in general, by default, they perform their function in the cytoplasm. Why? Because the mRNA, after it is transcribed, after it is completely processed, it comes out of the nucleus in the cytoplasm where it has to start the translation process. So before starting the translation process, these siRNA go and they bind to the mRNA and degrade it. But what it can also do, that these siRNA can go inside the nucleus and then they can block the expression of the gene before it is transcribed, before it comes out of the nucleus. Then and there it blocks the expression. Okay. So is it clear to you now, Madhvi? Is it clear to you? Okay, fine. Now, so now as we talked about that genomic imprinting where one of the allele is silent. So if the gene, so a gene has two alleles, one allele comes from mother, other allele is coming from father. So one of the allele is silenced. So the expression is only because of one of the alleles. This is your genomic imprinting. So basically, there are two mechanisms of genomic imprinting. There is one insulator model and there is one LNC RNA model. LNC RNA means long non-coding RNA. So long non-coding RNA means which are more than 200 nucleotidal length. These are long non-coding RNAs. So this is what is important for us to study, that non-coding RNAs have a role in genomic imprinting, especially long non-coding RNA, LNC RNAs. And this is a recently evolved and a more commonly utilized mechanism. This LNC RNA mediated mechanism is a more common mechanism. So what happens? It employed for non-coding RNA mediated imprinting at the IGF2R locus. You have to remember this locus, IG2R locus. Okay, so basically, the gist of this slide is that there are two methods of genomic imprinting, and RNC RNA model is the one which involves non-coding RNAs, which is our interest right now. So now we come to lecture 4. So we have 7 lectures. After lecture 4, we'll solve some questions and then we'll quickly see the other lectures and we'll solve the questions. Okay. So I'm again and again repeating that lecture 6 and lecture 7 mostly has examples. Examples of different diseases we shall see that. So We cannot explain each and every example here, but I'll try to cover most of the examples. You have to go and you have to understand that by yourself. And I hope that you have already read I mean, at least once you have gone through the lectures. Anyways, so now we talk about genomic imprinting. So, how genomic imprinting or how long non-coding RNAs helps in genomic imprinting. This is one of the examples of this. So, what this example says that during embryonic development, okay, when the embryo is developing, then some of the transcription factors which are very important for the embryonic development are Hox genes. If you have studied a little bit about developmental biology, you know about Hox. So Hox genes which are present within the Hox clusters. Okay, just a minute. Yeah. So these Hox genes, which sorry, yeah. So these Hox genes are important for the embryonic development, which are present in the Hox clusters. And what are these Hox genes? These genes control the body plane, that means anterior to posterior axis of the body. So these Hox genes are the genes which are very important for the embryonic development because they control the anterior posterior axis of the body. And remind you that we are studying the example of long non-coding RNA in genomic imprinting, and this is one of the examples. So this Hox gene is required for body plane, for head to tail formation. Now these Hox genes are modified or regulated with the help of some long non-coding RNA, which are known as hot air long non-coding RNA. So during development, Hox genes are regulated by long non-coding RNAs, and these long non-coding RNA, which is known as hot air, they are also encoded within the Hox gene cluster only. That means if this is the Hox gene cluster, within the Hox gene cluster, you can see in Hox G you have hot air. This is responsible for genomic imprinting. This is responsible for um, Hox gene regulation, and how it does, we'll see it. See, I'll just explain you that at a time, either Hox C should be expressed or Hox D should be expressed. If both are getting expressed, then there would be a problem. Okay, they will not, then the characteristics that you will get, that would be neither of Hox C nor of Hox D. That means at a time, either Hox C should be expressed or Hox D gene should be expressed. So if Hox C is getting expressed, then the expression of Hox D should be silenced. Now, how the expression of Hox D is silenced? With the help of these long non-coding RNAs, which are hot air. So hot air, they work in trans. What is the meaning of trans? Can anybody tell me the difference between cis and trans? What is the difference? Cis and trans? If I say a gene works in cis, and I say a gene works in trans. What does it mean? At least in this context, if you can tell me what cis and trans means. No. So if this long non-coding RNA, it acts on the same gene in which it is present, that is 6. But because it is not working on the same gene, it is getting produced from here and it works on a different gene. That is, it is working on a HOX-D gene. It is repressing HOX-D and not HOX-C. HOX-C. So it is working as trans here. So you can see in the next slide. Hmm. You can see over here. I'll just magnify it for you for just in time. Hmm. That's a lot. Hmm. You see, this hot air has trans regulation. How it has a trans regulation? Because it hot air is produced in HOX-C genes, but it acts on HOX-D gene. From here, it is acting here. So it is not acting within the same gene, it is acting on a different gene. Therefore, it is a transregulation. So what happens? Hot air represses the expression of HOX-D in the HOX-C expressing segment. That means where HOX-C has to express, there HOX-D should not express. So it represses the expression of HOX-D. Okay. And this happens by the genomic imprinting of the HOX-D. This regulation is trans-regulating regulation. That means the HOX-C cluster, but it is expressing and going to influencing the transcriptability of the HOX-D cluster. That means this hot air long non-coding RNA, although it is produced in HOXY, but it is going to HOXD and it is silencing the expression of HOXD. So it is going to a different gene. So it is a trans regulation. So what happens? That the clusters where HOXC has to express, there hot air will come and it will silence the expression of HOXD. This is the gist. Rest all is the details that you can go through, but this is the gist. This is the main concept behind it. Okay. Now, now we know that this hot air. Just a minute. Okay. So now we know that this hot air is responsible for repressing the expression of HOX D. But what happens if this hot air is mutated or it is knocked out? Then what will happen? Tell me. If hot air so what is hot air? Hot air is a long non-coding RNA. If this long non-coding RNA is knocked out, then what will happen? Huh. So that Keshwari, you have told it completely correct that cis is within the gene. Correct. Now tell me, this question is for everybody, that if hot air long non-coding RNA is knocked out, then what will be the consequence? What will be the result of this? What will happen? Think and try to relate to what we have just discussed. Nobody is able to answer? Okay, fine. So see, this hot air long non-coding RNA, it is responsible for the repression of OXD gene. But if this is only knocked out, then what will happen? There is no hot air. That means there is no repression of OXD. That means OXD is not getting repressed. That means it is de-repressed. De-repressed means it is not getting repressed. So there is gene de-repression of OXD which will take place, and this will result in homeotic transformation, which is a deleterious consequence. Okay. So, if in the mouse, targeted deletion of hot air, it causes de-repression of HOXD. Now, because hot air is responsible for the repression of HOXD gene. If hot air is only mutated or knocked out, then there is no repression of HOX-D gene. That means within the HOX-C cluster, now HOX-C is also getting expressed and HOX-D is also getting expressed. And this leads to homeotic transformation and malformation. So, what is homeotic transformation? That means whatever structure it has to form, it is not forming that structure, it is forming a different structure. If it has to form, you can see how the malformations are taking place. If it has to form six vertebrates or it has to form six vertebrates in this order. There would be some problem and it will form either more number of vertebrates or lesser number of vertebrates, or their sequence is different. There is some kind of malformations that happen. So that is why the repression of HOX-D with the help of this long non-coding RNA, which is hot air, is so important. Okay, because if it is not repressed, if it is de-repressed, then homeotic transformation occurs. What is homeotic transformation? The change in the structure with another structure. So, at a given place, a structure should be there and that is missing, instead, another structure appears. Okay. So, is this clear to everybody? What I was trying to tell? So, basically, what we read? We read that long non coding RNAs play a very important role in genomic imprinting. And one of the examples, this was explained by taking one of the examples of HOX gene. Okay, so HOX gene is responsible for the body plan of the embryo, which controls the anterior to posterior axis. And at a time, we all know that during embryonic development or any kind of development for that matter, whatever has to express, only that should express, not other factors should come and start expressing at the same time. Otherwise, there would be malformations. So, at a point, either HOX C should be expressed or HOX D should be expressed. Within the HOX C expressing area, HOX D should not be expressed. And how it is controlled? It is controlled with the help of a long non-coding RNA, which is known as hot air. So this hot air long non-coding RNA, it is produced, although it is produced in HOXC, it goes to a different gene, that is HOXD, and it silences HOXD expression. So now what is happening? Because HOXD expression is silenced, then only HOXC is expressed, it is fine. But if this HOXC is knocked out, then both HOXC and HOXD are getting expressed, and then there are homeotic transformations and malformations that occurs. Is it clear? Is it clear or not?
Yes, ma'am.
Okay, fine. So now we'll try to solve some questions. So the first question is, homeosis refers to formation of wrong structures, formation with broken structures, formation with big structures, formation with malformed structures. So think, and then in after 1 minute, I'll switch the screen. Okay. So Janara has answered D. Tarkeshri Madhuri has answered D. Kanchana has answered D. So everyone is correct. So homeosis is the formation with malformed structures. Here only we saw that malformation is occurring. Homeotic transformation results in malformation. Structures are forming, but they are malformed. Next is dicer. Dicer acts in the one should be nucleus, cytoplasm, or nuclear pore complex, where dicer acts. This we have studied in our previous class also, mostly in previous class only. Dicer acts in, Okay, I'm switching the screen. Okay, so Tarkeshwari, Madhavi, and Kanchana, they have answered C, which is correct. So dicer acts in the cytoplasm. We have already studied that in the cytoplasm, dicer acts and then it cleaves and it forms the non-coding RNA. After the RNA is formed, when it comes out of the nucleus to the cytoplasm, at that point only all this happens. Okay. Next question is hot air is a structural RNA, coding RNA, non-coding RNA, or a sponge RNA? Think. Okay, now I'll switch the screen. Okay. So Kanchana, Jahanara, Tarkeshwari, everybody has answered it correctly. So yes. Okay. What you have answered? Ha. Okay. Everybody has answered C, na? Yeah. So hot air is a non-coding RNA. Just now we studied that hot air is. So in non-coding RNA, can you be more specific? Which kind of hot non-coding RNA is? Hot air is which kind of non-coding RNA? To be more specific, LNC RNA. Good. So hot air is a long non-coding RNA. Fine. Then homeosis is the result of mutation in any gene. True or false? Homeosis is the result of mutation in any gene. Think whether it is true or false. So Tarkrishri is saying true. Janara is also saying true. Okay, I want more participation. अभिलाषा वो आइस ले आई हो वो आइस लाके उसको लगा देना हाँ ठीक है मैं फिर से अनम्यूट कर रही हूँ सो homeosis is the result of mutation. The answer is false. Now, why it is false? Because homeosis is there is no mutation that is happening. The only thing that is happening is that a gene which has to be expressed, the gene which has to be repressed, it is also getting expressed. For example, in this case, we wanted that HOX-D, it should not be expressed. But HOX-D is getting expressed. So there is de-repression. That means hot D was not getting repressed, it is getting expressed. Now, how it is getting expressed, that is a different scene. But the ultimate thing is that HOX D, which has to be repressed, it is getting de-repressed. And because it is getting de-repressed, it is getting continuously, it is activated. Therefore, there is expression of both HOX-D also and HOX-C also. So when both HOX-D and HOX-C are getting expressed at the same time, then homeosis is occurring and malformations are happening. So there is no mutation. There is no mutation in the HOX-C that is happening. There is no genomic imprinting that is happening. And because there is no genomic imprinting that is happening, both of them are getting expressed. Is it clear to everybody? Basically, genomic imprinting is getting, there is no genomic imprinting that is happening. So both HOXD and HOXC, both are getting expressed. There is no mutation that is happening here. Okay. I hope this is clear to everybody. Now, now we come to lecture 5. Now we'll talk about the different non-coding RNAs and their roles. From here, everything is about examples and examples. There are so many examples. So it's already 118. We cannot take all the examples. Otherwise, it would take the whole, I mean, I don't know. I mean, it will take 2, 3 hours even more to explain me all the examples with the pace with which I explain. So I'll try to get you a gist and I'll try to explain most of the examples. And I hope that with my explanation, you will be able to study one or two more examples that are given in your lectures. So non-coding RNAs. So now we know that non-coding RNAs have role in genomic imprinting. Then they are involved in RNA interference. They are involved in other kind of RNA silencing also with the help of miRNA. So is that it? No, there are even more roles of non-coding RNA. And what are those roles? Those roles are that first, they are required for muscle differentiation. So non-coding RNAs are required for muscle differentiation. What does it mean? That different stages of muscle development require different sets of transcription factors, and these different sets of transcription factors are regulated by different kinds of non-coding RNA. Okay, you can see from this figure. I'll try to, you see here, from progenitors, they are forming they are going through different stages: determination, myoblast, myocytes, myotubes. These are the different stages of muscle differentiation. And in each stage, you can see so many different kinds of transcription factors are important. In the progenitor stage, you can see PAX3, PAX5 is there. Then here PAX3, PAX5, MYF3, MYF5, myod. So there are so many different transcription factors that are required for the transition of one stage to another stage. And these transcription factors are regulated by different sets of microRNAs. For example, you can see microRNA 206. This microRNA 206, it represses PAX5 and eutropin. So what happens? PAX5 is responsible for the self-renewal ability. Now, when PAX5 is getting inhibited, then no self-renewal will occur. That means now it will move towards the differentiation process. If proliferation is inhibited, then it will move towards the differentiation process. That means these microRNAs have different roles, which ultimately helps in either the proliferation or differentiation or attaining the specific features during muscle differentiation. You can see here, MIR206, it is inhibiting PAX5, and PAX5 is required for cell renewal. So that means MIR206 is inhibiting cell renewal. That means it is helping in differentiation. Similarly, MIR31, MIR1, MIR133. So these different microRNAs have different roles, which ultimately helps in the differentiation of muscles. This is one example of non-coding RNAs. Then non-coding RNAs are also required for retinal development. Okay. So what we are studying? We are studying the different roles of non-coding RNAs. So we have already studied that non-coding RNAs are required for genomic imprinting. We have studied that non-coding RNAs are required for RNA interference or silencing. And now we are saying the other different roles of non-coding RNA, in which we have studied first that they are required for muscle differentiation. And the second role is that these non-coding RNAs are involved in retinal development. So you can see these are the three factors: VAX2OS1, 6,3-OS, and RNCR2. So these are the three factors which are regulated by non-coding RNAs. You can go through it, it's just a theory. Then non-coding RNAs are also required, are also responsible in some kinds of diseases. Okay, so you can see that microRNAs, these microRNAs are involved in cancer. Just a minute, what I'll do is, um, let me put it here. Hmm. So you can see that non-coding RNAs are response are have roles in complex diseases such as cancer, Alzheimer diseases. So in cancer, what happens? That this CPG island is hypermethylated. Okay? And when non-coding, this hypermethylation is because of the non-coding RNAs. But when these non-coding RNAs are inhibited, then there is hypermethylation is removed. And when hypermethylation is removed, then what happens? Then the transcription of genes takes place, and this transcription of genes ultimately leads to tumor formation. So in this way, you can see LNC hot air. This already we have studied in Alzheimer disease, also some other. So basically, all the non-coding, most of the non-coding RNAs have role in complex diseases, as in they control the formation of those diseases. Here MIR200 is preventing the tumor formation by repressing the transcription of some proto-oncogenes. But as soon as this MIR200 is removed, these proto-oncogenes are expressed and it might lead to the formation of tumor. Okay. So first role is in muscle differentiation. Second role is in retinal development. Third role in complex diseases. Okay. Now, now we'll study from here. We'll try to study the role in complex diseases in a little bit more detail. Detail as in things are all the same. The mechanisms you can just go through and read. I'm just telling you that what is the gist and what is the main logic behind it. So now these non-coding RNAs, specifically the long non-coding RNAs, they are with the help of these long non-coding RNA, some kind of cancer progression is also controlled. That means these long non-coding RNAs, they control cancer progression. Okay, these long coding RNAs control the cancer progression. And how it controls? There are two examples of this. First is H19, and this H19 is regulated in bladder cancer and in colon cancer. So what happens? Upregulation of this maternally imprinted LNCH19 due to loss of imprinting occurs in a wide range of metastatic tumors. So basically what happens? That this LNC RNA, which is H19, it is genomic imprinted. But if a gene which is imprinted, a gene which is silenced, it is also expressed, then that leads to the formation of a lot of tumors. So that is what is the case. And then again, there is another example in colon cancer, where what happens that this H19, it serves as a precursor for miRNA 675, which targets the tumor suppressor. So these are all the mechanisms. You can read these mechanisms, but I tell you that there is, I mean, even if you're not going into very depth of the mechanism, that's fine. You just need to know that these long non-coding RNA, they regulate the cancer progression, and they regulate the cancer progression in bladder cancer, in colon cancer. The example is H19, and you can remember the points that are bold in, they are bold. Going in depth and learning the mechanisms, if you have really interest, you can go. Otherwise, you can just have a gist of it. Okay. Then there is another example which is KCNQ Q1OT1. So what does it do? That this KCNQ1OT1, it is required for normal heart development. Okay. So how it regulates the normal heart development? By imprinting of certain genes in the early stages of cardiac development. Okay. So if the imprinting is not proper, then there would be formation of some kinds of cancer. So this KCNQ1OT1, it silences neighboring genes. So in the early stages of development, it silences in the early stages, it silences some genes that are its neighboring genes. But if there is down regulation of this KCNQ1OT1, then this silencing is stopped. And when this silencing is stopped, all the genes are getting expressed, like whatever genes that has to be silenced, all those genes are getting expressed. So what happens because of that? There is aberrant 3D structure of chromatin, which causes abnormal contact with the enhancers, and therefore cell proliferation occurs, and therefore cancer occurs. Okay, so one important point that I can explain you, if this is a gene, there are certain promoters and there are certain enhancers. So enhancers, it enhances the expression of promoters. So if there is no genomic imprinting, what happens? This promoter and this enhancer, earlier they were not in contact with each other. But now, because of this loss of genomic imprinting, because this KCNQ1OT1 is not present, this enhancer and promoter are now in close proximation because there is aberrant 3D structure formation. Now, the 3D structure that is formed is so aberrant that this promoter and enhancer has come close to each other. And now this promoter is getting even more expressed because this enhancer is enhancing its expression more and more, more and more. So, more gene expression is there. So, there is more cell proliferation, which is leading to cancer because now there is uncontrolled growth. Okay, there is uncontrolled growth. So you can see that just one long non-coding RNA, that is KCNQ1OT1, it can create so much of devastating differences that if this KCNQ1OT1 is down-regulated, then it will not silence the genes. If the genes are not silenced, they will be expressed more and there are aberrant 3D structures. If there are aberrant 3D structures, then these enhancers, which were earlier very far away from the promoter, they have come into close proximity. So now this enhancer is enhancing the promoter so much that there is cell proliferation, which is ultimately leading to cancer. Okay, so this is another example. Then next example is Terra. So what is Terra? So basically, you should understand that telomerase is the enzyme which is required for the repairing of the ends of the telomere. So it with every DNA replication, the telomere ends get shortened. With every round of DNA replication, the telomere ends are getting shortened. So to maintain the telomere's length, telomerase enzymes comes into action. But although telomerase is required, it is a very important and a very essential enzyme because it maintains the telomere length. At some point of time, when the cell has to undergo for death, then the telomerase must be shortened, and at that time, the telomerase activity should be inhibited. So this TERA is a long non-coding RNA which inhibits the activity of the telomerase. If this TERA is down-regulated, then what happens? Telomerase activity is very high, enabling the cancer to evade apoptosis and subsequent programmed cell death, and it will keep on growing and multiplying and multiplying. Okay, so this is also very interesting. So, because each and every cell after a certain stage it has to die. So, it has to undergo apoptosis, it has to undergo senescence. So, one of the mechanisms to undergo apoptosis or senescence is shortening of the telomeres. And this shortening of the telomeres can only be achieved when the enzyme which adds more telomeres at the end is inhibited, and that enzyme is telomerase. So this telomerase is inhibited with the help of a non-coding RNA, which is TERA. So if TERA is not there, telomerase will keep on adding the telomeres to the end. The cell can never die. Cell becomes a cancerous cell. So you can see that these long non-coding RNA are so essential that if they are not present, they are mutated or they are down-regulated, it can lead to the formation of different kinds of cancers. Okay, now we'll see the next one. Mm, hmm. Now again there are other examples of long mechanisms of long non-coding RNA, which ultimately leads to cancer progression, such as P10P1, then you have malate1, and then you have zist1. So these are the three examples that again describes the cancer progression because of the down regulation of the long non-coding RNA. You can just read it. It's nothing much to explain here. So now one important thing that we might be studying in our next lecture also are the telomeres. So telomeres are the, if this is your sequence, at the last you have telomeres. And these protect the chromosome from replication related loss of vital genetic information. Okay. So these telomeres are very important, and these telomeres were first described by Elizabeth Blackburn, and who won a Nobel Prize for it as well. And what are these telomeres? These are nothing but the repetitive DNA sequence at the ends of all human chromosomes and other eukaryotes. So at the ends of the sequence, at the end of the DNA, you have telomeres. They contain thousands of repeat sequences. So the important property or the important characteristic of these telomeres is that first, that these telomeres are present at the ends of the DNA sequence, and second is that they have a lot of repetitive sequences. Okay. So in humans, there are 46 chromosomes and thus 92 telomeres. Why? Because in each chromosome, two telomeres is there. One at one end and then another at another end. So if 46 chromosome, that means 92 telomeres are there, one at each end. And these telomeres protect the chromosome from the replication related loss of vital genetic information. Okay. Now we'll just move to the questions. We try to solve them. So the first question is, which of the following RNA do not contribute in human diseases? miRNA, siRNA, rRNA, or lncRNA? Think. Think, and then I'll switch the screen. Okay, I'm switching the screen now. So, Janara has answered C, Anjana has answered C, and Tarkeshwari has also answered C. So let's see. rRNA. Okay. Okay, so lncRNA we have seen, miRNA we have also seen. Now we come that siRNA or rRNA. So most of the students have answered, I mean all of them have answered rRNA, but the correct answer is B, that is siRNA and not the rRNA, because although you have not studied anything much about the rRNA, this thing, rRNA related diseases, but there are a lot number of diseases, human diseases that are caused because of rRNA. I'll just give you one or two examples. So it is not mentioned over here, but or if it is mentioned, then you have to again check. But I'm just telling you that there are some kinds of diseases which are known as, if you have heard, I don't know whether you have heard or not, it is known as mitochondrial myopathy, okay, and some other kinds of syndrome, such as myotronic dystrophy, and some other diseases, some kinds of cancers, okay. These are just a few examples. These are actually well-known diseases which are caused because of rRNA. For miRNA, for lncRNA, we have already studied a lot of them. But for siRNA, not much is known. So here the answer is siRNA and not rRNA. Okay. So there are some, okay. So I think some students have answered. Yeah. So the answer is B and not C. Okay. Next question is, non-coding RNAs are not involved in development, differentiation, transcription elongation, and imprinting. Think. I'm giving you 1 minute and then I'll switch the screen. Okay, now I'm switching the screen. Let's see. Just a minute, I have to check this also. Hmm. Okay, so let's see what students have answered. Okay. Uh, so two students have answered, Kanchana and Jahanara, both have answered C. And C is the correct answer. Yes. So non-coding RNA, they are involved in imprinting. We have studied so much. They are involved in differentiation. We just studied that in this slide. We studied that here they control in muscle differentiation and they are also controlling the proliferation. So non-coding RNA, they control proliferation also and they control differentiation also. They might help in the start or stop of the transcription, but elongation they do not do. They might help in transcription initiation, such as shRNA, um, not shRNA, but one example we have studied in the beginning, I think, where they were helping in promoting the transcription. Here, but that helps, but here it is helping not in transcription, it is helping in translation, and that too initiation. Okay, so that means transcription elongation, nowhere it is helping. So the correct answer would be answer C, that is transcription elongation. Now comes the next question. shRNA do not participate in chromatin condensation. True or false? shRNA do not participate in chromatin condensation. Think. Okay, so I'm switching the screen now. Okay, so SHRNA did not participate in chromatin condensation. So Momin Jahanara has answered false. Kanchana has answered true. Okay. So the answer is false. Why it is false? So what is the function of shRNA? Can anybody tell me what is the function of shRNA? It helps in the silencing, right? It helps in the silencing. So it silences the gene so that transcription does not happen in the nucleus. So when it is silencing, when shRNA is involved in silencing during genomic imprinting, then because of silencing, what will be the state of chromatin? After silencing, what will happen to chromatin? Whether it will be condensed or it will be very much separated and elongated. What do you think should be the position of chromatin? The chromatin should be condensed because when a gene is silenced, then chromatin is so much condensed and densely packed, which forms a heterochromatin region. Okay. So that means shRNA, they do not participate in chromatin condensation false because they do participate in chromatin condensation. And how do they condense the chromatin? By silencing the gene. With the help of silencing, they condense the chromatin. Okay. Next question is sense and antisense transcripts often act antagonistically. Antagonistic means opposite. Think 1 minute and then I'll switch the screen. Okay. So, Jahanara and Momin both have answered true, and the correct answer is true. Yes. So that means obviously sense and antisense have opposite behavior all the way long. We have been learning about this only. They are complementary to each other and they are opposite behaviors. Fine. Next is RNA interference was first discovered in zebrafish, Saccharomyces cerevisiae, Saccharomyces pombe, or C. elegans. So I think okay, I have not included that information. So I'll just see if you know. Otherwise, I'll explain you. I will tell you. Think and then I'll tell you. RNA interference was first discovered in anybody? Okay. So Tarkeshwari is saying C. elegans and it is the correct answer. Fine. Next question is genomic imprinting is seen in all living organisms. True or false? Genomic imprinting is seen in all living organisms. True or false? Okay, I'm switching the screen. Okay. Kanchana has answered is false and Janara has answered as true. So the correct answer is false. Why? Because see, always remember in all living organisms, if an organism is haploid, then what will happen? Can it have genomic imprinting? Tell me, if an organism X cell is haploid, Can it have genomic imprinting? No, because it should be diploid. One allele should come from father, other allele should come from mother. That is the first requirement. Only then you can have one of them as imprinted. If there is only one allele, then how it can be imprinted? This is one of the reasons. Second of the reasons is that not all the cell of the body show the phenomena of genomic imprinting. There are specific genes at specific time points, especially during the embryonic development. Okay, just genomic imprinting is very prominent, especially during the embryonic development. So not all the time all the genes are showing the phenomena of genomic imprinting. And secondly, that not all the organisms are diploid. Some are haploid also. The haploid cells cannot show the genomic imprinting. So it is not seen in all living organisms, but still it is a very important phenomena that controls the expression of genes, and hence it helps in preventing the progression of many kinds of diseases, including cancers. Okay. Now, next question is, hmm, the RNA interference mediated gene silencing is an ATP dependent procedure. True or false? If you have information, you can answer it. Okay, I'm going to switch the screen now. Let's see. Hmm, okay, yeah. So it is correct. So RNA interference is an ATP dependent process. That means it is energetically expensive. Now, next question is, if the siRNA is a complete homologue of the target mRNA sequence, what is the net result? Double helix formation, translation inhibition, chromatin modification, or mRNA degradation? Try to answer the question. First, read the question carefully and then answer. Okay, so now I'm switching the screens. So, Tarkeshwari has answered as C, which is chromatin modification. Okay, anybody else? Okay. The correct answer is mRNA degradation, which is sixth. Sorry, D. Why it is so? See, we have already studied here. I have explained it two, three times. Hmm. So, if there is complete base pairing, then what happens? The target is cleaved. If the base pairing is not complete, then target mRNA is not cleaved, but your translation is repressed. Okay, so that means here you can see that siRNA, it forms a perfect base pairing. That means there is perfect complementarity between the siRNA and the target mRNA. And because there is perfect base pairing that happens, the target, that is your target, is your mRNA molecule. Okay, your target is your mRNA molecule, and this mRNA molecule is cleaved. Whereas in the case of miRNA, you can see that the base pairing is not complete. It is an incomplete base pairing that happens, and therefore your target molecule, which is again an mRNA molecule, it is not cleaved, but your translation is repressed. So what is asked? What is there in the question? Let's see. Question says, if siRNA is a complete homologue of the target mRNA sequence. That means is a complete homologue of the target mRNA. That means if this is your target mRNA and it is completely able to bind with this target mRNA, then what will happen? Then mRNA would be degraded. Okay. Now next question is the last question. Which of the following RNA can induce gene silencing? miRNA, snRNA, ssRNA, or ncRNA? Again, in this question, hint, you might think that there are more than one correct options, but try to look for the more specific one which is more specific. Hmm. Okay, so I'm switching the screens now. A. So Madhvi has answered, Tarkeshwari Madhvi has answered A, Janara has answered D. Kanchana, what do you think should be the answer? Let me see the options. A and D. So I told you these are the only two options where you can confuse. Kanchana has answered A. Okay. Now you see snRNA cannot be the answer. ssRNA cannot be the answer. We have two candidates now, miRNA and ncRNA. What is ncRNA? What are ncRNA? These are the non-coding RNAs. And what is miRNA? miRNA is a type of non-coding RNA which is specifically involved in gene silencing. So miRNA is a more specific answer rather than a non-coding RNA because non-coding RNA can be of many different types. snRNA and ssRNA are also non-coding RNAs only, but they are not involved in gene silencing. So here, if nothing would have been given, if these three, if it the options would have been like tRNA, mRNA, rRNA, and ncRNA, then you would have picked non-coding RNA as the option. But here, since non-coding RNA is also given and miRNA is also given, and miRNA is a more specific answer, so you have to choose miRNA and not non-coding RNA in this case. So always look for the more specific option amongst the given options. Fine. So now we'll just quickly revise. Hmm. So here we have studied about, first we have studied the role of YY1. So YY1 is required for the localization of the cyst in the nucleation center. And then we have studied in the last class, as we have studied about dosage compensation, we studied that inactive X chromosome harbors most of the mutations during tumor. You can see this is a normal cell, this is a cancerous cell. In normal cells also mostly mutations are in the X chromosome only. In a cancer cell also, most of the mutations are occurring in the X chromosome. In the X chromosome also, mostly in the inactive X chromosome. Then we studied about genetic imprinting, that genetic imprinting is mediated with the help of shRNAs. And yeah. Then we studied that this shRNA can bind to the gene and inhibits its expression or silence it with the help of two ways. First is a DNA recognition model and RNA recognition model. Okay. Then we studied that inhibition or regulation can also happen at the level of translation, and that is known as RNA interference. And then we studied that RNA interference. No, normally we know that RNA interference is responsible for the silencing of the gene expression, and non-coding RNAs are involved in the silencing of the gene expression. But in some cases, it can also activate the gene expression, as in this case, where double-stranded DSR ARNA, when it binds, it clears the ribosome binding site, and therefore translation can take place. Then we studied that about RNA interference, what are the different steps and what are the different functions of RNA interference. Then we studied the difference between miRNA and siRNA. siRNA has a perfect base pairing, whereas miRNA does not have a perfect base pairing. Okay. Then we studied that RNA interference not always happens in the cytoplasm. It can happen in the nucleus also, and where the siRNA will act as shRNA and it blocks the transcription process. Then we studied that there are two models for genomic imprinting, insulator model and LNC-RNA model, and mostly we are focusing on the LNC-RNA model. Here we studied that how long non-coding RNA, and the name of the long non-coding RNA is hot air, and how this hot air non-coding RNA controls the expression of HOX-D by acting trans. It's a trans regulation. It is produced on HOX-C, but it acts on HOX-D. This we have studied. Then we studied that if this hot air long non-coding RNA is knocked out, then there would be homeotic transformation and malformations will occur. Then we studied that apart from the previously mentioned role, non-coding RNAs have some other roles also, such as they are involved in muscle differentiation, they are also involved in retinal development, they are also involved in some complex diseases such as cancer, Alzheimer diseases, and some other kinds of cancer progressions we have studied. Then there is TERA, which inhibits the activity of telomerase, which will, if telomerase is expressed more, then there would be no stop in the telomere addition, and then it might lead to cancer progression ultimately. Then we studied what are telomeres. These are basically the repetitive sequences which are present at the ends of the chromosomes, and if they are present at both the ends, so if there are 46 chromosomes, that means there are 92 telomeres, and they protect the chromosome from replication related loss. So vital information is not lost during replication because telomeres are present at the end. Okay, so now we'll stop the class. If you have any questions, you can ask me. Anybody has any questions?
Homeosis, one second. Homeosis.
Okay. So basically, you should understand. So, mm, where it is? Hmm. You should always understand that when embryo is getting developed, okay, so there has to be a perfect way, a perfect defined way in which your organs develop. So if a particular gene X is responsible for, let's say, it is responsible for the development of five fingers. Okay, I'll just explain you very briefly. If a particular gene X is responsible for the development of five fingers, and there are two copies of this X, one has to be repressed, otherwise, ultimately, you will have 10 fingers. Each will produce five. But this X is getting imprinted with the help of some non-coding RNA. But if this non-coding RNA is deleted, then this X is repressed or not? No. This X is activated. Now this X is also there, this X is also there. So now you have in one hand, normally you should have five fingers. In one hand, now you have 10 fingers. So what is this? This is malformation. And this malformation is known as homeosis. That means particular gene, it whenever there is a development of embryonic development, particular gene should be expressed only up to a level it is required. If it is expressed more, if more than one genes are expressed simultaneously, then you will have abnormal growth. And that abnormal growth will lead to a malformed body, and that is your homeostasis. Is it clear?
Yes, ma'am. It caused by mutation, ma'am.
It can happen because of mutations also. So that is the C. It can happen because of n number of reasons. This is one of the reasons by which it can happen. It can happen, suppose, if this X gene, which is responsible for the development of five fingers, if this is mutated, there is some problems, then you will not have proper finger formation. You may have lesser number of fingers, or you may have thumbs like this, which are like joint thumbs, more than one, like you see in some of the peoples. So it can happen because of mutations also, and this is also one of the reasons. Okay?
Thank you, ma'am.
Okay. So now we'll stop the class. I hope you were able to understand something, and we'll meet in the next week. So bye.